Hi, I'm Ramin Sakhtemani. I'm a graduate student in Bhagwat lab here in the Department of Chemistry of Maine State University. Our lab is interested in biochemistry of DNA and a group of enzymes that act on DNA. Recently, we published a paper on mapping genomic uracils. So DNA uses 4 different bases, adenine, guanine, cytosine, and thymine, to code all the genetic information, while uracil is not a normal base in DNA. There is a chemical reaction called cytosine deamination that can convert cytosines to uracils, and this happens at a low base rate all the time, while there are a group of enzymes that can catalyze this reaction. We have several of these enzymes. They are part of our immune system, and their primary purpose is to target and mutate viral genomes when they enter our cells. While if they are unregulated and overexpressed, they can also damage the host genome. In fact, the mutational signature and the overexpression levels of these enzymes has been detected in multiple types of cancer. So it's very important to understand these enzymes and how they act and their mechanism of action. In order to do that, our previous graduate student and the co-first author of this paper, Dr. Vimukthy came up with this idea and developed this uracil pull-down method. It starts with extracting genomic uracils and sonicating it and cutting it into small pieces, and then through a couple of chemical and enzymatic reactions, putting a binding tag on those fragments that have uracils, and then use that tag to pull down and enrich those fragments, use it for next generation sequencing, and get the sequencing data. In order to establish this method, I use human apoBac3A, one of those cytosine deaminases, in E. coli cells. I express that, and then I use the pull-down method to pull down and sequence the targets of apoBac3A. I used several genomics and bioinformatics tools, and I used the university's supercomputer or GRID to accelerate this computation. I wrote a little algorithm to take the sample sequencing data and take a negative control, combine that, and remove all the biases that may arise from DNA pull-down or DNA sequencing, and finally detect those targeting regions in the genome. One of the results that came out of this study was that we found that this enzyme is targeting regions of the genome that code for tRNAs. Our collaborator, Dr. Mike Lawrence, looked at human cancer data sets and found that in cancer data that have the mutational signatures of these enzymes, tRNA coding genes are also heavily mutated. So something that we found in our study was also true for human cancer data sets. The next step for this study can be to use this method for mammalian systems and find where in the human cells these enzymes are causing damage, or changing the structures of these enzymes and get more insight on how different structures can cause different types of targeting. Thanks for watching, and if you want to know more details, there is a link in the description.